Hello and welcome, my dear friends. Hope you all are doing well today. So, in my last video, we had already discussed about genetic material and its packaging in prokaryotic and eukaryotic organisms. So, let's start today with DNA replication process. What DNA replication means? When DNA duplicates and form its own replica is known as replication. The DNA molecule regulates and controls all the activities of the cell. When the cell reproduces, the DNA also should replicate itself to contribute to the daughter cells. As a carrier of genetic information, DNA has to do two important functions. One is heterolytic function and second one is autolytic function. Hetero means when DNA directs and synthesizes some other chemical molecules like RNA and proteins. And autocatalytic function, when DNA synthesizes its own replica. That means it's a DNA replication itself. So the process by which DNA duplicates itself is called DNA replication. And in eukaryotic organisms, replication of DNA takes place only once in S phase of interphase in the cell cycle. Cell cycle we had already studied in 11th standard. So in S phase of interphase, only that phase where DNA replicates. So let's see the semi-conservative mode of replication of DNA. What is semi-conservative means? Semi means when one strand remains same and the replica on another strand will form. That means it's a semi-conservative replication. This model was proposed by Watson and Crick on the basis of anti-parallel and complementary nature of DNA strands. This process is known as semi-conservative replication because two copies of the original DNA molecules are produced. Each copy contains one original strand and one newly synthesized strand. So there are few steps involved in the semi-conservative replication. Number one is activation of nucleotide. So how this activation of nucleotide happens? So there are four different types of nucleotides present in DNA. That is DAMP, DGMP, DCMP and DTMP in the nucleoplasm of the cell. So they are activated by ATP, adenosine triphosphate, in the presence of some enzyme which is known as phosphorylase. So that is the process of activation of nucleotide happens. Then second point is the point of origin or initiation. And that begins with a specific point which is O, that means origin, and T is a termination point. So that start with O and T, and at the point of O, the enzyme endonucleases that cut the strand of DNA for temporarily. So that is how it start the origin of the unwinding of DNA. Then third step is unwinding of DNA molecule, where the enzyme DNA helicase operates by breaking weak hydrogen bonds of those DNA strands. Then the DNA strand separates and unwind and they form a Y-shaped replication fork. So that is the process of unwinding of the DNA. Then fourth step is the replication fork. So that the point formed due to the unwinding and separation of two strands of DNA, like they form a Y-shaped fork. So that is known as replication fork. That is known as replication fork or replicating fork. Then the step 5 is the synthesis of new strands. So it began with the help of some RNA enzyme that is known as RNA primer, and these nucleotides, those molecules, they bind to the complementary nucleotide on the template strand by forming hydrogen bonds. So that these new bonds or new strands they start synthesizing. Then sixth is leading and lagging strand. So the template strands with free 3' end is called leading strand template, or the 5' end is called lagging templates. So the process of replication always start with 3' end. So it has to be remembered that the process of replication always start with the 3' end of the template strand and the process ends towards the 5' end. Then the formation of daughter. So the DNA synthesized on lagging template take place in the form of small fragments they call Okazaki fragments. The DNA primer are removed by DNA polymerase and replaced by DNA sequence with the help of DNA polymerase 1. So finally DNA gyrase is a topoisomerase enzyme that form the double helix to form a daughter molecule, and then the formation of daughter DNA molecules happens. So at the end of the replication process, two daughter DNA molecules are formed, and in each daughter DNA, one strand is parental and another strand is newly synthesized. So you can say that the 50% contribution is by mother DNA, hence it is known as semi-conservative replication of the DNA. So here in this picture you can see there is a unwinding happens. So after unwinding, the strands will become a Y shape, then lagging strand and RNA primer, and then Okazaki fragments are formed. So it starts from 3' and it's end at the 5' end. So this is a whole structure of semi-conservative replication of DNA, where you can see the one strand is a mother strand and another newly synthesized strand is formed, and that is one strand, one DNA is forming two daughter DNA molecules. So that is known as semi-conservative replication of the DNA. Then next is about protein synthesis. The proteins are very important biomolecules. They serve as a structural component, enzyme and hormones. The process of protein synthesis that includes as a transcription and translation. So transcription, when DNA to mRNA form, that process is known as transcription, and from mRNA when it is forming a protein or polypeptide, that step is known as translation. So this whole unidirectional flow of information from DNA to RNA to mRNA and to mRNA to the proteins is known as central dogma of molecular biology. So once it is been asked what is a central dogma, so from DNA it is forming mRNA, the process is known as transcription, which happens or which occurs in the nucleus, and from mRNA to the polypeptide, this process is known as translation, that occurs in the cytoplasm. So this whole process is known as central dogma, and it was given by F.H.C. Crick in 1958. So let's elaborate the process of transcription. So during transcription, information of only one strand of DNA is copied into RNA, and that DNA strands act as a template enzyme, RNA polymerase, that catalyzes the formation of RNA transcripts. So three steps are involved: initiation, elongation and termination. So by the name itself you can identify initiation, where the process starts, the transcription process starts; elongation and the termination, where the process of the transcription ends. So each transcribed segment of DNA is called transcription unit, which consists of a promoter, the structural genes and a terminator. The promoter is located towards 5' end of the structural gene. The RNA polymerase binds to the specific promoter. Structural gene, two strands of DNA have opposite polarity. The DNA dependent RNA polymerase catalyzes the polymerization in direction which is 5' to 3' directions. So the DNA strands having 3' end to 5' end polarity act as the template strand. The information on this strand of DNA is copied on mRNA, and this is called a sense strand. And the termination or terminator is located at the end, which is 3' end of the coding strand, that is downstream, and it is defined as a end of the transcription process. So in this diagram you can see very clearly there the process of transcription starts from 3' to 5' end, where one is a promoter and second one is a terminator, and above one is a template strand, and this is structural gene and this is a coding of strand. So this is a transcription unit where transcription starts. So after binding to promoter, the RNA polymerase moves along the DNA and cause local unwinding of DNA duplex into two chains on the region of the gene. So only one strand function as a template, which is antisense. One only strand will work as a template, and the other strand is a complementary or a coding strand, which is also known as sense strand. So the ribonucleoside triphosphate that joins to the base of DNA template chain and the transcription proceed, the hybrid DNA RNA molecule dissociate and the mRNA molecule that will be free. So this is how the process of transcription happens, and the transcription process where DNA to mRNA forms. So I hope in this video it will be able to help you in understanding the process of replication with the semi-conservative mode of replication, and also we are able to understand the process of transcription. In my next video we will study about the process of translation and some